Hello, we are here again to talk on microbial examination of water. And before this, if you remember, we have talked the various aspects of the fresh water contamination, either that is river, either that is a stream, either that is any other source, how that contaminates, how we can protect that water reservoirs. And here we have to talk that how we can check the microbial load in water. So we are talking on microbial examination of water. We are checking water sample for the microorganisms. That microorganisms are present, there are not. So here it is our outline agenda, waterborne disease, pathogen indicators, coliform, Streptococcus, Enterococcus, enumeration method. In enumeration method, we will talk about membrane filter technique and MPN multiple tube fermentation and surface water industrial standards. We will talk about this, that which type of standard has been developed for this, that how much microbial load is acceptable that and beyond that is not acceptable. How much water can wild bird contaminate, that only human beings are not responsible, but there is a huge population of birds on this globe. They are also contaminating the water bodies, and how we can estimate that microbial contamination load in water. And at last we will state that how microbes survive in environment and how they are getting access to the water bodies. So waterborne pathogens in 1991 cholera epidemics, thousand uh, thousands cases and 10,000 deaths happened. So we know very well that microorganisms, viruses, protozoa, helminthes, spirochetes, rickettsia, and algae, these are all our microorganisms. These are present in environment, and from environment to healthy individual they are coming through water. So it was the fact established in 1991 due to cholera epidemic that pathogens which are present in environment getting access to healthy individual through water. And these are some cases which happened through water's initial epidemics January 1991, August 1991, February 1992, and November 1994. These are the epidemics which has been spread through water because water is the most abundantly using substance in human life, and it has been estimated that more than 80% disease, communicable disease, are transmitting from infected or from patients to healthy individuals through water. Bacteria are present in environment in it. Pathogenic bacteria and non-pathogenic bacteria all are present in environment and getting access to healthy individual through water. Viruses are present in water and getting access to healthy individual through water. Protozoans are also present in environment and getting access to healthy individuals through water. Similarly, helminthes, spirochetes, rickettsia, and algae also getting access through water to human body. Bacteria Bacteria are causing enteritis, diarrhea, and dysentery. And the most bacteria which are responsible for this are Campylobacter, cholera, Escherichia coli 0157H, Salmonella, Shigella, these are causing enteric fever, Salmonella causing typhoid and paratyphoid, some are causing paralysis, and some are causing botulism. Bacteria, eye, ear, and skin, they are also causing eye, ear, and skin infections, and numerous type of bacteria are involved, so we can say miscellaneous bacteria are involved. Bacteria also causing infection that is called urinary tract infection UTI, and mostly E. coli and other microorganisms are involved in it. And if you will see, viruses are also causing enteritis, diarrhea, and dysentery, and microviruses involved in rotaviruses, Norwalk viruses, flu-like liver damaging viruses, which are causing hepatitis A and hepatitis E, and some are causing paralysis. And the example of paralyzing virus is poliomyelitis. Protozoans, these are different type of protozoans. These are also causing disease in human, and these are Giardia, Cryptosporidia, and amoeba. These are getting access through water to healthy individual and causing different disease. And if you will see the helminthes, roundworms, tapeworms, hookworms, and whipworms, these are present in environment in various stages, in variant forms, in egg form, in two-cell form, and in advanced larvae, and it is embryonated egg are digested through water to human, and these are causing infection and disease in human because these are taking nutrients from healthy individual and causing negative health effects on host, intermediate host. So if we will see the most of the either these are bacteria, viruses, and protozoans, and helminthes, and others getting access to healthy individuals through water bodies. And other, if you will see, algae, microcystis, dinoflagellates, and algae, these are present in environment and consumed by and getting access to healthy individual through water, fungi, water-related disease, and which are causing malaria in water-related disease where different reservoirs our hosts can grow and causing malaria, schistosomiasis, yellow fever, and dengue fever. These all diseases are caused through water. So if you will see most of the pathogens which are present in environment getting access to healthy individual through water. If water is not treated before consumption, any microorganism, viruses, algae, protozoa, worms can get access to healthy individual and causing negative impact. So because water is the most abundantly using substances, it is used for drinking, it is used for washing, clothing, bathing, for every purpose of the life. If water is not pure, that is biologically contaminated, that can cause severe health effects on on people. So what is the problem? Because if we have seen there are numerous waterborne pathogens. So for all to check all these microorganisms, viruses, fungi, protozoa, helminths in water, it is not possible, it is difficult. Because there are many bacteria, many viruses, many fungi, many protozoans, and too much. Numerous waterborne pathogens are there. And in a laboratory, for one sample, we cannot check all these bacteria or pathogens. And another problem, individual pathogen number may be too low to detect in a reasonable sized water sample. Suppose for analysis we are taking 1 ml of water sample, maybe that pathogen is not present in that 1 ml and it is present in other remaining water. So there are the huge chances of the escaping of pathogen from a small water sample. Therefore, it is also another big problem. Isolation and detection of some pathogen can take several days, weeks, or months. So for one sample, we cannot check all these things because there are various microorganisms, uh, bacteria, viruses, fungi, which are taking too much time to be isolated, to be detected, and to be identified. So it is again a big problem. For one sample, we cannot do all these things. And maybe some laboratories and some institutions or organizations have no facilities for detection of all these pathogenic microorganisms. And another thing, cost is also increasing. Absence of one particular pathogen does not rule out the presence of another. If one microorganism is not present, that we cannot say that all pathogenic microorganisms are not present. Maybe others are present for particular one we have tested. Maybe that is not present, and others are present. It also will cause problems. So therefore, to solve this issue by keeping these all problems in mind, that pathogens are numerous. Individual pathogen may be at low number and cannot be detected in a reasonable size of sample. Isolation, identification, and detection of one pathogen is not possible because some are taking too long time, and absence of one microorganism uh, cannot rule out that others are also not present. These are uh, expensive and very costly, and no one can afford, no one organization, no one laboratory, no one individual can afford all these things. So keeping these problems in mind, it has been established that we have to go for the indicator organism concept, that we will use one indicator. If that microorganism is present, it means others are also present. If this organism is not present, means others are also not present. Therefore, by keeping these all problems in mind, people have gone for the indicator microorganism concept. What is indicator organism concept? That correlated to the presence of pathogen. If that microorganism is present, maybe others are also present. If that particular microorganism or indicator organism is not present and others are also not present. Population large enough to isolate in a small water sample. Because in indicator microorganism, the population of indicator microorganism is too large enough to isolate in water sample. In a small water sample can be detected easily even in 100 ml. And this indicator organism concept is rapid, and it is very cheap, inexpensive. It is safe, not culturing pathogens, that is normal flora of human intestine and non-pathogen. Therefore, it is safe for handler, for transporter, and for grower. It has no negative effects. And that is indicator organism concept. Which type of microorganism we are taking as indicator? That is coliform group, total coliform group. Coliform group belongs from Enterobacteriaceae, and these coliforms are facultative anaerobes, can grow in absence of water as well as can grow in presence of water. Facultative anaerobes, and these are gram-negative rods. These are unable to form any spore, therefore we are saying these are non-spore formers. These are rod-shaped, These have ability to ferment lactose and produce acid, gas and acid within 48 hours at 35 degrees C. So this group is Enterobacteriaceae is indicator organisms, indicator concept of indicator microorganism. We are using coliforms as indicator because these are numerous in number and can be detected in a reasonable water size sample easily. These are not pathogenic. Coliform genera indicates Enterobacter, Klebsiella, Citrobacter, and Escherichia coli, which is normally called E. coli. These are total coliforms, and coliform group is considered as indicator group. If any member of coliform is present in water sample, it means all pathogens may be present. If coliforms are not present, it means no pathogenic microorganism is present. Why? Because coliforms are huge in number. These can be detected in water sample. And one thing more I have to add over here, coliforms are surviving for longer time than other microorganisms in water sample. Coliform group. This is total coliform, fecal coliform, all total coliform criteria that grows at 44.5 degrees C. Suppose first we are checking total coliform which have ability to grow at 44 degrees C and fecal coliform uh, which can grow at 44.5 degrees C and other coliforms can grow at 37 degrees. Escherichia coli individual species, this is enzyme specific. Suppose first we are checking total coliform, in second step we are checking, first we are checking total coliform, then we are checking fecal coliform, and lastly we are checking E. coli, which is individual species and it is enzyme specific. E. coli is normal flora of human alimentary canal. If E. coli is present in water sample, means all pathogens may be present. If E. coli is not present, it means water is not fecally contaminated and other pathogenic microorganisms must not be present. Therefore, E. coli and fecal coliform are considered as indicator group, and these organisms are surviving longer time compared to other microorganisms in water bodies. Streptococcus and Enterococcus are also present in intestine. Fecal Streptococcus, Streptococcus faecalis, Streptococcus faecium, S. avium, S. bovis, S. equium, and S. galerinerum. Enterococcus, which are present in Enterobacteriaceae, fecal streptos that survive in 6.5% sodium chloride, as fecalis, as faecium, as avium, and as galeninerum. And due to this, we also can select Enterobacteriaceae family from water samples. So now we are coming how we have to check the water sample, how we can analyze the water sample. Maybe we have to keep up to this and analysis method we will talk in next step. So I'm little bit talking again on indicator microorganism. Enterobacteriaceae, member of Enterobacteriaceae is E. coli. E. coli is the member of Enterobacteriaceae. Okay, because this is present in human alimentary canal and present in feces. If water is contaminated with feces, uh, that must contain E. coli. If E. coli is present, that indicates the water is contaminated with feces. If water is contaminated with feces, maybe all type of human-related pathogens may be over there. And for detection and isolation and identification of other microorganisms, there are too much problems. Therefore, we are selecting on the E. coli to check. If E. coli is present, means water is fecally contaminated. If water is fecally contaminated, that may contain and harbor all types of microbial strains in it, which can cause disease in humans. If E. coli is not present, water is not fecally contaminated, and other pathogenic microorganisms which are human related must not be present in that. Therefore, we are going for the concept of indicator microorganism group. So, uh, for the detailed study, you must have to go through our book, and you can find the suitable this procedure in detail from the Prescott Microbiology, which has been recommended to you. Thank you. Have a nice time.